This is part 2 of the chapter 13 lecture notes. There we go. All right. So let me blow this up a little bit if I can. I can't use a pen when I do that, but I can use the hand. Okay. So this is what the results of meiosis look like when independent assortment occurs if the genes are on different chromosomes. So what we're looking at here are two fruit fly genes. One is represented by a G and the other is represented by an L. It doesn't say in the diagram, it doesn't say what the G or the L stand for. There are several traits that fruit flies have, eye color, their wings. They either can have long wings or they can have these short vestigial wings, is what they're called. They just have different traits like humans do. So I'm not sure what the G and the L stand for, but you can see in these chromosomes that the top homologous pair of chromosomes, the one on the left has the big G version, the big G allele, and the one on the right has a little g allele. And then the chromosome on the left, of the bottom set, has a big L allele, and the one on the right has a small, a little L allele. So what we see in the gametes are that um, we get all the possible combination of G's and L's. So each gamete, each egg or sperm is going to have one G and one L. So it can have a big G and a big L, um, a big G and a big L, a little G and a little L, or a little G and a little L. When independent assortment occurs, it can also have, in other words, if these chromosomes line up differently during metaphase 1, they line up in this order, where on the bottom pair the X is on the left, the red X is on the left. Okay, then you can get big G little l and little g big L. So these are all the possible combinations that you can have in the offspring after fertilization. You can end up with a fruit fly that's all heterozygous for both traits, big G, big L, little l. You can end up with a fruit fly that's homozygous recessive for both traits. You can end up with a fruit fly that's heterozygous for the G trait and then homozygous recessive for the L trait. Or you can end up with recessive for the G trait, homozygous recessive, and heterozygous for the L trait. So in other words, if the main lesson here is that if the genes are on different chromosomes, then independent assortment is going to occur, okay? But if they're on the same chromosome, that doesn't always happen. Independent assortment doesn't always happen. So if genes are on different chromosomes, you're going to have independent assortment of those traits. You're going to have all the possible combinations in the sperm and the egg, that all the possible combinations of alleles. But if you look at genes on the same chromosome where no crossing over occurs, there's no crossing over, there's no exchange of pieces of chromosomes, then you don't see that. You don't see as much variety. All you're going to see is big G in the sperm or egg is big G, big L, or little g, little l. And so you don't get as much, uh, you're not going to get as as much variety in the offspring. So you're either going to get offspring number 1 or offspring number 2. You're either going to get an offspring that's got big G, big L, little l, or you're going to get an offspring that's homozygous recessive for both traits. And if you do have crossing over 100% of the time, you're going to get the same results that you got here. You're going to get the same amount of variety as what you get when the genes are on different chromosomes. But what normally happens is crossing over does not occur 100% of the time. So in Morgan's 1912 experiment, he actually tested thousands of fruit flies, and he found out that where the parents were heterozygous. Okay, so he tested the offspring of 2 fruit flies that were heterozygous for both traits. And he found that 965 of the offspring ended up, I'm saying this wrong, I'm so sorry. One parent was heterozygous, the other parent had only the recessive. So it was, these are the results from that. So what you get is you get 965 of the offspring are heterozygous for both traits. 944 are homozygous recessive. And this is these are the results that you would see normally if there's no crossing over. But since there is some crossing over, we're going to have a few that have the other possible combinations that you get from independent assortment. Big G, little l, and little g, big L, little l. So the point is you don't have 100% crossing over would mean that all these numbers would be equal. But since you don't have as many, 206 and 185 is what you have of the second 2 possibilities of offspring. And since you don't have as many, we know that crossing over is happening, but not at 100% frequency. Okay, so the frequency of crossing over helps us to understand where the genes are located on the chromosomes. Okay, so on this slide, it shows crossover and homologous recombination during synapsis of meiosis 1. Okay, so we're looking at 2 traits, the A trait and the B trait, and we're noticing that on the original blue chromosome, there's big A, big B. But after crossing over, you end up with a piece of the green chromosome and a piece of the blue chromosome. So in that way, you have one chromosome that has big A, little b, and one that has little a, big B. And then you also have a green chromosome that is like the original, just little a, little b. So this is what you're going to see. This is the result of crossing over, is you can have a chromosome with a big A, big B, big A, little a, big B, or little a, little b. And the 2 in the middle are called recombinant chromosomes. So crossing over gives you all the possibilities of combinations of alleles in the offspring. But when crossing over doesn't occur, then you only get the top and the bottom versions. All you get without crossing over is this or this. Okay? So they used the frequency of crossing over to map the chromosomes of the fruit fly. They used those crossing over frequencies to show where the actual genes were located on the chromosomes. So they found out that the gene for Arista was located in one position on the chromosome. The Arista, by the way, just for your information, are little bristles on the end of the antenna. So they can either be short or long. And they found that the gene for body color, black or gray, is 48.5 units down from that. And cinnabar eyes or red eyes, or the eye color gene, is 57.5, is at position 57.5. And wings, whether they're vestigial wings or normal length wings, are a little further down. And then brown eyes are at position 104.5. I'm sorry. Whether or not they have brown eyes or red eyes is at position 104.5. So, so the genes for these different traits have been mapped based on their frequency of crossing over. The higher the frequency, the closer together the genes are. Okay, so in other words, the vestigial wings or normal wings and red eyes or brown eyes, these two genes here, they're going to be, they're going to have more, they're going to have less crossing over because they're closer together. But the Arista gene and the brown eye versus red eye gene are farther apart, so they're going to have more crossing over. Okay, here's a karyotype, and you can tell that it says it right here. I was going to ask you, but pair number 23 are both X's. So this is a human female. It shows all 23 pairs of chromosomes. And what we can see from this karyotype or any karyotype is, especially if you look at, I like pair number 7 because you can see the bands really well. Homologous pairs are going to they're going to be the same height. They're going to have their centromere in the same position. So here and here is where the two sister chromatids are attached, the centromere. And they're going to have the same banding pattern because those bands represent different genes on the chromosomes or different pieces of DNA. So the banding patterns are going to match on homologous chromosomes. And what that tells us is that those genes, they are genes for the same traits. They don't have to be the same version of the trait, but they're genes for the same traits. If this one at that position has, if that gene is for eye color, then so is the gene on the other homologous, the other chromosome of the homologous pair. Okay, now something that can happen, this is a totally different topic from what we've been discussing. This hasn't got anything to do with linkage or crossing over, but non-disjunction is something that can happen during meiosis, and it is the failure of chromosomes to separate. Now, they can fail to separate during meiosis 1. If they fail to separate during meiosis 1, then the homologous pairs stay together, they don't segregate. Now, if they fail to separate during meiosis 2, it is the sister chromatids that don't separate. And you can see that happen here. But what you end up with is sperm or egg. In other words, the gametes are going to have either an extra chromosome or they're going to have one less chromosome than they're supposed to. So each of these should be they should be haploid. Each of these gametes should be haploid. But because of non-disjunction, some of the gametes are missing a chromosome and some have an extra chromosome. Something else you can see from this picture is this during meiosis 2, if the non-disjunction happens in meiosis 2, then 2 of the possible gametes will not be affected. So if non-disjunction occurs during meiosis 1, all of the gametes are affected. Okay? Down syndrome is a disorder that results from non-disjunction of pair number 21. So when the pair number 21 ends up being a triplet instead of 2, you have 3 chromosomes for pair number 21. That's called trisomy 21, and the offspring will result in Down syndrome. And this is just showing you the correlation of frequency or risk of Down syndrome in the child and mother's age. And of course, the risk goes up significantly when the mother is in her late 30s and 40s. And as she gets older, the risk just becomes higher. And this video is on slide 18, and it just helps you visualize. This is using, it looks like Twizzlers and Reese's Pieces or something like that, little candies. So it's another way of illustrating meiosis, kind of like we did with the beads. And next lecture we will talk about, the next section we'll talk about